Okay, yeah, there we go. Beautiful. Beautiful, beautiful, beautiful. Welcome everybody. Thanks for being here. I apologize, a little bit late tonight. The Facebook, we hit 6,000 members in the Facebook group, so we did a little special stream for that, and it ran a little bit long. So yeah, welcome back. Happy Monday. uh, Last night we were supposed to do the weekly stream last night on Sunday. We're gonna go back to doing it on Sundays. We've had a lot of ice and a lot of like rain, I guess, in the area that's been causing Spectrum, our Wi-Fi provider, a primary Wi-Fi provider, to lose service like intermittently for a lot of people. So last night I just didn't have Wi-Fi. I didn't want to do it on my phone because I couldn't display all the stuff that we need to display. So we're here tonight. What's up, David, Sean, Matt? Steve, Bobby, what's up? Gilberto, how you doing? Trujillo, yeah. What's up, guys? All right, let me get into the zone here. We're gonna do some nutrients. I'm gonna jump right into it because we got a lot to talk about tonight. I am going to, first of all, we have a drop coming very soon. It's as far as we know right now. It's gonna be exclusively on Multiverse, at least for a while. And that's going to be our Jehovah's Witness crossed with iced latte, and we're going to be announcing a name for it, and that's going to be available exclusively through Multiverse, and it is, I've requested photographs on the Facebook. I really want to try to post as many, like customer images of this strain as we can, because we released it as a freebie like a while back. We gave decent number of them away, so we're hoping that a good number of people have pictures of that strain to post, and we can use your pictures on the website. We can send them over to Multiverse. They can use them. You can have your picture featured on one of like the biggest, if not the biggest, autoflower seed banks out there. So I thought that's pretty cool. Jolly Man, I'm going to talk about it here also. I kind of made that graphic for YouTube for this stream, and Ice Witness. Ooh, that's pretty good. Bobby, it might be it. You know what? Bobby Stanley gets credit. I think Ice Witness, or a play on that, but Ice Witness, that's pretty good. It's like witness protection, We already have that. I really like that. That's good. Simple but good. Yeah. Okay. I think we might call it that. If we do, Bob, you get a free packs. I'll send you a whole multi packs worth 12 seeds. I'll send them to you for free because you named it. That's good. That's a good one. I like it. So, okay. All right. Well, we're gonna talk about two things tonight. First of all, we are going to talk about, finish our nutrient schedule and our nutrient stream. That's what you called it, Sean? Nice. Post pictures for sure, Sean. Frozen Witness. That's good too. I like Ice Witness is like, like you killed a witness. Like, oh, we iced them. I kind of like that. It's funny. Frozen Witness is good also. But Unwelcome Visitor. That's good too, yeah. So, okay, well, if anybody can top Ice Witness, then maybe you'll get your. But for right now, that's the winner in my head. So I can already think of like the graphics we can make for it. It's pretty good, yeah. Okay, we're gonna finish our nutrient stream tonight. So we're gonna finish the early veg, late veg, early flower, late flower numbers. Or we'll do as much as we can within the time frame where it makes sense. And we're going to talk about those numbers. We're going to talk about those PPMs. And I'm also going to talk about plant tissue analysis and what it is and why we do it and what it does for us. And I'm also going to talk about filial generations. This is a discussion that I've been, from recently, I've been getting more questions about this. And I think it's just because we're getting more people who are new to speedrun, and they, a lot of new growers too. They haven't been with us since the beginning and haven't heard me yap on YouTube and Facebook for hours and hours, like probably 100 hours at this point of live streams talking about random shit. Yeah, Sean, text them to me or something, or email, we'll figure it out. Frozen Jehovah, that's pretty funny too. I Ice Jesus. Anyway, yeah, we'll figure it out. But yeah, definitely Terp Sneeze is a good one to post for pictures. Also, Jehovah's Latte. It's funny also. Okay, yeah, we're finishing that, doing that, and then yeah. So I've had a lot of people asking about filial generations. I've seen a lot of people mentioning F2, kind of implying that it's more stable than F1. And I just wanted to clear some stuff up. So I made a little graphic that I thought kind of might help people understand some stuff. And I didn't really get to go through the whole thing on Facebook. So I'm going to go through it, probably, hopefully, maybe like a little bit of like an abbreviated version tonight here. Let me see if I can shrink this window down. And let's do it. Let's do it to it. Okay. So I have here off to the side, let me shrink me, real tiny me, even smaller. Okay, so I have here off to the side a graphic that I made very quickly. That is not complete yet, but we will post this so you guys can download it or whatever. If you guys want us to make like, a kind of a poster or something along these lines, we can do that too. But okay, so I'm going to talk about genetics here. So I'm sorry, I'm dragging this around the screen so much. Let me just get it kind of situated. Okay, we're going to talk about genetics here and what makes, genetics more stable, what makes them less stable, what the filial generations mean. There, I'll put myself right there. And we're going to talk about just why certain genetics, or not certain genetics, but, you can have a strain that's an F3 and another strain that's an F3 or an F4 or an F5, and one's significantly more stable than the other. And I'm going to talk about why that is and what contributes to that real quick. Okay, so for me, on my, stage left, my right, stage left, you see an F1 cross that says homozygous dominant. And on my stage right, you see heterozygous dominant, or just heterozygous F1. So real quick, talk about the differences here. Homozygous dominant, each plant receives a version of a gene that's responsible for a specific trait, one version from each parent. So they're called alleles, alleles, and they determine what shows up in the phenotype. Some can be dominant, some can be recessive, and they are not always expressed in the phenotype. Only one of them will be expressed in the phenotype. So what you're seeing here is called Punnett squares, and you might remember those from biology class in high school, maybe, or college. But homozygous means, homo means the same, zygous referring to the alleles, and then heterozygous means different, also referring to the alleles. So what you see here is two capital P's means dominant purple gene, dominant purple allele, dominant purple allele. So two dominant purple alleles. And then you've got your two uppercase G's, two dominant green alleles. And then in the heterozygous version, you have an uppercase P and a lowercase g, and an uppercase G and a lowercase p. So these are heterozygous plants that are less stable than the homozygous ones. So if I were to take two plants, cross them together, and they are stabilized plants, then we get what we like to call a stable F1. Okay? All of our F1s, I would say, all the ones that we've released are stable F1s, which means that the two parents have been stabilized to an extent, to a degree, where we're comfortable crossing them together. And what we're trying to get is an F1 generation that has an even blend of traits from both parents. And that's what you see here on my stage left, where you've got all four squares are the same half purple, half green. and they're all PG. Now, when you go down and look at the resulting generation that happens, you might see that they don't all look exactly the same. And the reason there is because, yes, they both have a dominant, co-dominant purple gene and a green, or sorry, purple allele and a green allele, meaning that they have shared traits between the two. So you get some purple and some green on the plant, but there in each individual there can be random genetic, just not mutations necessarily, but genetic variances. So some of the dominance can be a little bit, the co-dominant isn't always 50-50 is the best way to phrase that. It's not always 50-50. A lot of times it is. Sometimes it's like 60-40, 70-30, where you've got a lot of one parent and a little of the other parent. They're both there in the phenotype. You can both see traits from both, but one of them is really dominant. So not, is more dominant than the other dominant one. So that's what you see here. So on the left of your screen, you see some plants that are PG, just like all the rest, but they're more green. And you see some plants on the right that are PG. Sorry, I'm talking about specifically the left column there. You can't see my cursor. So the heterozygous co-dominant. You know what, that where it says heterozygous co-dominant under the one, that's not supposed to be there. I got to fix that. I kind of just copied and pasted and then changed some stuff. But anyway, yeah. So on this left column here, I'll put I'm going to just put this as the only one that you're seeing. Why can't I click on myself? There we go. So what you're seeing here are on the left here, you've got plants that are a little bit more green. On the right, you've got plants that are a little bit more purple, but they're still a mix of green and purple. And in the middle, you've got nice 50-50 purple-green. So now, if you were to cross your... Oh, that's another thing that needs to be fixed. I don't think I sent over the most recent version of this, actually. Yeah, there's some mistakes here. So just in the fact that these squares... the one that says PP on the top left should be completely purple, and the one that says GG on the bottom right should be completely green. I forgot to change that. So regardless, when you cross two plants that are in your F1 generation together, you cross a PG with a PG in this case, you get plants, you get more variation. So here we've got one genotype across all of them. They're all PG. All co-dominant PG. That's F1. Moving into F2, you end up with three separate phenotypes here. A PP, a GG, and two PGs. So now we've got, you can see, there's four PGs in the middle there. The four middle plants are on the top and bottom are all, this is eight individuals from each, in this generation. So you've got in the center there, you got all four PGs. One of them's a little more purple, one of them's a little bit more green, but they're all PGs. They're all half green, or some green, some purple. And then on the left, you've got your pure purple, and on your right, all the way on the right, the two on the top and bottom are your pure green, co-dominant, homozygous purple, co-dominant, homozygous green. So now what could happen? You could cross two completely purple, co-dominant, homozygous plants together, and you would end up with a completely purple, more stabilized F3. So this is your F3 generation here. Now, if you were to cross two plants, now the reason I chose those two is because one of them's completely green, PG, and then one of them is, GG, and then one of them is mostly green with a little bit of purple. And if a breeder doesn't really notice or is not that, isn't that attentive to what they're really seeing, I'm just going to get myself out of the way here. Hold on. So if the breeder is not super attentive to what they're actually seeing there, you're going to notice that, there's some plants that are just like almost completely green, but with a little bit of purple. And then there is a purple allele hiding in that genotype. So if you were to cross it with a completely green plant, instead of getting, all green offspring like you would if you crossed the two completely GG plants together, you end up with a 50-50 split here, where you've got half PG and half GG. So you've stabilized it more, but it's not as stable as if you had crossed two of the exact same plants together. And that's what the struggle is. Hold one sec. Okay. That is what the struggle can be. Make myself a little bigger here. With breeding, is that you might not always choose, even though the phenotype might look very similar, the genotype might not be. And you've probably heard, if you've heard our live streams, if you heard me talk, or if you read any of my posts, you might hear me say we redid a generation. We redid the F3. And that could be because we chose a plant that we thought was really similar, but in that genotype, there was some extra goodies that we weren't anticipating, and the next generation didn't come out as stable as we wanted. The F3 generation just wasn't stable enough. And we'll redo it. We'll go back to the F2, we'll grow it again, we'll cross two F2 plants together that have a better chance of giving us a stable, more stabilized F3 generation. And that is, not a lot of breeders will read. Sometimes they'll just choose the plants that are the most similar, cross them, and give you an F3. And it might not be as stable as it could be. And same thing with F4, F5. If you were to choose, if I were to go buy a 3-pack of Granite Haze and plant 2 of them and reverse one and cross it with the other one, if they were the exact same phenotype and the genotype was really consistent between the two, then you're going to get yourself, if it was a Granite Haze F4, you're going to get yourself your own homemade Granite Haze F5, and it would be pretty stable. But if you have 2 phenotypes that are kind of where the genotype is pretty different, 2 plants where the genotype is pretty different, then those were probably not the 2 ideal plants to cross to make an F5. And your F5 is going to be inferior to the real F5 that we have because we put the work into finding the individuals that are similar enough, and we crossed a bunch of different pairs of individuals, and we chose the ones that were the most stable to sell as the Granite Haze F5, the most consistent. So that is where the quality of a filial generation comes into play, because there are factors that can make one F5 not the same, not as good as another F5, or not as stable at least. Okay, so then briefly, that's why you can see you've got a good F3 and a bad or not as good F3. And then briefly, I'll talk about this here. Heterozygous plants, it gets even more complicated. And the reason, this is a very simplified version of cannabis genetics. Just like people, just like most other organisms out there, short of like, fruit flies and green peas and stuff where it can be really simple like that. You've got, those are the examples that you learn in school when you're talking about Punnett squares like this. They use Mendel's peas, Mendelian genetics. That's where Brother Mendel was a monk back in like the 18th century, I want to say, that invented this Punnett square. That's not, well, I don't know if he invented the Punnett square, but he invented Mendelian genetics and the idea of alleles and that kind of stuff. He's the father of this type of genetic theory. And so Brother Mendel, he was a monk, and that's Brother Mendel is why he named his company after that, if you guys know about that genetics company. But anyway, so heterozygous is more complicated because, each of these plants, so the one on the left here, let me do it like this. The one on, let me go get myself back too. I should probably look this way. But the one here on the left, the PG, is a purple plant with a dominant purple gene and a purple phenotype, but it has a recessive green phenotype. And then the GP is a dominant green phenotype, but has a recessive purple phenotype. So if you cross those two together, then you end up with a PG codominant, and then you end up with a PG recessive, they're expressed, but it's 2 recessive genes that are being expressed together. And then you end up with a GG, which is a dominant and a recessive heterozygosity, I believe is what, yeah. And then you end up with a dominant and a recessive purple. So you end up with, instead of being like it is on the other side, 2 dominant purple genes to make a purple plant, you end up with a recessive and a dominant purple gene, and a recessive and a dominant green, and a recessive, 2 recessives and 2 dominant. So if you were to cross this lowercase GP with this uppercase PG, the uppercase is always going to win out. So you're always going to end up with those being expressed. So when you have two recessive traits or two recessive alleles being expressed, that means when you cross that with something else, it's going to be really weak. And the other plant that has more dominant genes will take over the cross, and it will you're not going to see those traits from that plant show up in the next generation because they're recessive traits that you're trying to put up against the dominant traits. Hopefully you guys are understanding this. I understand it might sound a little bit confusing if you've never heard of any of this stuff before, but yeah. So I wanted to talk about that. I slapped together that diagram real quick because I had a bunch of conversations about that in the past week or so, and I just wanted to kind of explain what all of that was and what it meant, and give you guys a visual to understand it a little bit. So let me pop over to the chat because I have not looked at that in a while, and let me see if there's any questions that need answering. Sean says Zaza Genetics preaches about this stuff all day long. I'll have to look into them. I've not heard of them. Do you cure in mason jars or do you use the different method? So Matt, for seed production, we don't do a lot of curing, but when we do flower production and for our clients that do flower production, depending on the volume, for you guys at home, jars are a great way to do it. We recommend like a little bit of a faster dry, and you're leaving your moisture level in the center, like the stalks in the center of the buds are a little bit higher moisture level than the outside. The outside might be a little crispy, but the inside has some nice moisture. You put that into your airtight container or your next to airtight container. You seal it up. You don't open it for like a week, and that moisture will balance out, and you'll end up with a nicely dried, really nice, fluffy, but still kind of soft, cottony bud. And that's our recommended drying. And then curing, you can just kind of keep it in that sealed container. Burping is not really something that if you do the dry right, you shouldn't need to burp. If you end up with a lot of chlorophyll still, occurring in your, it's overpowering your terpenes, then burping does make sense to kind of to refresh the air in there. But if you did your dry right, burping is not as necessary. What speed run strains have the highest probability of turning out purple? Blackberry Haze, Pound Dog, and then the new Grand Exchange. Those three, I would say, have pretty good. And then there's other. Those are like where you have like an actual genetic purple plant. And then we have other strains like God Particle, Rainbow Six, which is coming back. Even Granite Haze, all those kind of have the affinity to have a green plant that turns a little bit purple. But Adam, what did you miss? I was just talking about filial generations, and I had a little diagram I was going over. Okay. Sean says you have the Pound Dog F4. Yes. Dead Folk says Pound Dog that has Gnome genetics in it comes out purple a lot. It does. So I've talked about this before. I'll mention it once more briefly. We don't breed specifically for color often at all. I mean, it's not something that's high up on our priority list for breeding. So when we're stabilizing strains, we could have two plants next to each other that are identical in every way, except one is green and one is purple. Terpenes are the same. Height is the same. Frostiness is the same. Bud structure, speed from seed to harvest, your vigor, your nutrient resistance or your nutrient tolerance. Everything can be exactly the same. But one's green and one's purple, not a big deal to us. We feel that we've done our job, and that stabilization is pretty good. We've got every other trait except for that purple. And the color of the plant doesn't impact the quality of the flower. I mean, if somebody's really looking for a purple strain, then yeah, okay, you could say it impacts quality. But in our minds, quality is more about all of those traits that are more than just visual. Frostiness, it's visual, but it also indicates that it's got really good trichome density, which means you've got, more resinous plant in a lot of circumstances. There's things that are visual that are important, but color alone is not something that we have high up. It's still on our list. It's on our list of breeding traits, but it's just not as important as other ones for us. Jollyman says, get a Canatrol. I've heard good things about the Canatrol. Cool. So if you have any questions about the filial generations, I can answer those if you come up with a question throughout the, and I'll make that graphic once I fix it. There's a couple mistakes on there. Once I fix it and make it public, I'll post it. I'll make it public. I think I can post it on the YouTube community thing. So, yeah. Okay. I'm going to go back. So it's just started 1 year ice latte, S3, and a mystery seed. Mystery seed is killing it right now, 19 days old. Thanks for hooking it up. Awesome, Taylor. Yeah, hell yeah. Definitely post some pics on whatever social media is and tag us if it's a tag one, or if it's on Facebook, post it in the group or something. Or the Reddit, post on the subreddit, r/speedrunseeds. But yeah, and then S3 just means it's a self, so like, in that example on that diagram, if one of those, where's my diagram? Here, let me move myself out of the way real quick. So if your PP on the left there is the dad and your GG on the right is the mom, the way we work with this is we just reverse like 2 or 3 branches from that PP plant, and we will reverse those branches, allow them to produce pollen, but not to the point where they drop the pollen, where they release it. As soon as they're about to release, we can kind of tell when they're about to release the pollen. We cut those off. We take them to another location, and we put them in a specific liquid solution, kind of like a flower vase, and we allow them to continue to grow, essentially, or mature, and they'll drop their pollen elsewhere, and then we'll come back and we will pollinate a full group of, let's say we're crossing PG with GG, then we'll have like 10 GGs, and we'll cross that pollen that we got from those branches with all of those, and we'll hit the same, the plant that those branches came from, we'll hit that plant as well. So it pollinates itself. So even if we're making like ice latte crossed with Jehovah's Witness, if we hit that ice latte with the ice latte pollen, then we've also made an ice latte S generation. So yeah, okay. Get rid of you, put me back up here. So let's get back to talking about nutrients. Where we left off last week, here. Sorry about that replacement since I missed out on final boss. Yeah, you have to remind me exactly. It's hard to keep track. If you're talking about seeds that are getting sent out, then awesome. Yeah, final boss. It's one of the best. Somebody just posted some pictures of it on the Facebook. It's such an amazing strain. I love that strain. Okay, so I believe I have our. Here we go. Document from last week. It's untouched. We're just going to kind of continue. So I'm going to go over a quick summary recap from last week. So what you're seeing here is the important things to know for creating an autoflower nutrient schedule. Guaranteed analysis, percent of each element. So what that means is the guaranteed analysis, which we can find in our pictures here. So here's your guaranteed analysis for the Flora series from General Hydro, and your NPK number, 501 for the micro, 216 for the grow, and 054 for the bloom. Those are your percentages of nitrogen, phosphorus, potassium. And then on the back of the label, they tell you all the same, and then they also give you everything else that's in there. NPK are what are considered to be your macronutrients, so that's why they're on the front, because they have to be displayed. But then on the back, it tells you all the other things that are in there percentage-wise, and it also tells you the where they're derived from. So having your guaranteed analysis tells you what percent of each element is in your nutrients. And then having your application rate, which a lot of nutrient companies provide in their feeding schedules. Your application rate is how many milliliters per gallon, if it's a liquid, of that nutrient that you are adding into your water how many milliliters you're adding into each gallon of water to make your nutrient solution to feed your plants. So having both of those numbers means that we can do some math to figure out the parts per million of each of those elements where you see the percents there, since we know how many milliliters we're putting in and we know how many milliliters are in 1 gallon total, that means that we can calculate, if it is 1% potassium, how many parts per million of actual potassium are in that gallon of water, or in each gallon of water when you're feeding your plants. And that is how nutrients are expressed in commercial agronomy. That's how nutrients are talked about when you're at growing any crop, really. Like you're talking about a farmer growing corn, you're talking about somebody growing wine grapes or hops. If they're feeding their plants on a large mass scale, then they're almost always talking about it in, if it's liquid nutrients like this, conventional liquid nutrients, they're talking about it in context of how many parts per million of each of these micro and macronutrients. So our example here is we know that 1 gallon of water equals 3,785 milliliters total. And then we know for our Flora Grow at the very beginning, we were adding 2.5 milliliters. So that means that if you take 2.5 milliliters and divide it by the total number of milliliters in a gallon, which is giving you the percent of your nutrient solution that is Flora Grow. So what percent of this nutrient solution that I've just made, this 1 gallon of nutrients, is actually Flora Grow? And this is your number here. This is not expressed in percents. You would want it would be 0.066% is what it would be. But we're looking at this as a decimal instead. And then now we know that 1% of a million is 10,000. So that means that 1%. So let's say 1% of a million ppm is 10,000 ppm, and a million parts per million is just 100% something. So like if you a Flora Grow is like how many parts per million of Flora Grow are in Flora Grow? A million. Like that's how that works. So we know that 1% is 10,000 parts per million. So we can do our math, which I can write out the formula here for you. So that means that your milliliters per gallon divided by total milliliters in 1 gallon, and then we're going to do, one sec here. I don't want to make this confusing for you guys. Okay, yeah, let's do it like that. So times I'll say it like this and I'll explain it. Okay. So when I say the percent integer, I mean like if it's 6%, you're not going to type in 0.06. You're going to type in 6. So that's where, I mean, if you guys can think of a better way to describe that number, that is what we're talking about there. But so your percent integer gets entered there. You multiply it by 10,000 because each percent equals 10,000, and then you're multiplying it by the number of milliliters in your gallon over the total milliliters in the gallon, which is going to be a decimal, and you end up with your ppms. And we did that for Flora Grow at the beginning, at the 2.5 milliliters per gallon, and we ended up with 13.2 milliliters of nitrogen, 6.6 parts per million of phosphorus, 39.6 parts per million of potassium, 3.3 parts per million of magnesium. And then we did it for Flora Bloom, and we did it for the Flora Micro. We started to do it for the Flora Micro. There we go. We did it for the Flora Micro, and we also added in the Cali Magic. So now we can go and look at the next stages. So if we were to go through and do every single growth stage here, I can take a look and see how many of them are. We wouldn't really have to calculate that many numbers. You guys tell me if you want me to. If this is something you feel like you already kind of understand, and we can move on to talking about the desired PPMs in a nutrient schedule, or if you want me to continue going on and calculating the. Here, I'll switch back to me. So yeah, if you want me to continue going on and calculating the each part per million, if you want to see me do it a few more times, or we can go and kind of just like, if you know the formula now, if you're comfortable with the formula, we can talk about applying it to what your desired PPMs are for any nutrients. So you guys tell me in the comments there. And, there used to be a good calculator for this online, and we are working on getting a calculator for it up. It's used to be a really good one, but okay, so let's see. Example 1A and 1B, total nitrogen equals 19.5% at 120 milliliters per gallon. You might be putting a decimal in the wrong place, Matt. Our PPMs, if you're looking at like nitrogen of the 1A, I think we're at quote me at like 210 or 190. I don't have them all memorized, but it's right around 200 parts per million. They're actually very similar to the numbers that I have up on the thing here. So, okay, I'll continue. Unless you guys want me to go back and go over calculating PPMs more, I will move on to talking about applying them to your actual, oops, didn't mean to do that. I meant to do this. Okay, so I'm going to go back here and we're going to talk about our early veg part per million numbers here. And you know what? I meant to pull up the study prior to this. Maybe I can I'll search for it on Google and we can pull it up together. But here's some recommended for autoflower recommended part per million input numbers for cannabis, for autoflower cannabis, and this is for early veg. So your nitrogen, you want it to be around 180 to 220 parts per million. Your phosphorus, 50 to 55 parts per million. Potassium, 170 to 190 parts per million. Calcium, 180 to 200. Magnesium, 60 to 65. Sulfur should be at least 20 to 50. But if you're using magnesium sulfate, your sulfur might be like a few hundred parts per million. The good news is that sulfur does not present as a toxicity problem almost ever, unless it's in such high numbers, along with a lot of other stuff being in such high numbers that the salinity of your solution overall ends up being really high. Then you start to see some problems. But sulfur, it's almost impossible with normal nutrients, not making any ridiculous mistakes, to have a sulfur toxicity happen. And that is a good news. It's good news when you're using magnesium sulfate because you've got more sulfur than you do magnesium in there. So in order to get the right amount of magnesium, you need a good chunk of sulfur. But it's also a very critical nutrient, and you want to make sure you have enough. So if you're not using magnesium sulfate, then you want to definitely add something that has sulfur in it, like your micronutrient sulfate salts. Like so there's a lot of your micronutrients, like zinc sulfate. Like manganese sulfate. A lot of those are the least expensive and most common version of them are in sulfate salts. A lot of nutrient companies use something called chelated micronutrients, which are micronutrients that are wrapped in another molecule, usually like EDTA. And that's what the molecule is. And it wraps around it and makes it easier for the plant to uptake. So it makes it less likely to kind of get stuck in the soil, you could say. So yeah, I mean, aside from that, so those might not be sulfates, and you might not have really any source of good sulfates in your nutrients, and that's when you have to think about adding some. But other than that, we're pretty safe with sulfur because usually it comes along with something else. So make sure I'm not missing nothing here. Okay, so yeah, let me switch back to this, and we're going to talk a little bit about the next stage here. Where's my streaming software? There we go. Okay, so let's just do this. And once we're finished with this document, I will make it available to you guys. Let's just move on to the next page for this. Okay, so we break it into 4 different growth stages: early veg, early veg, late veg, early flower, late flower. And we're going to talk about late veg now. So we're going to bump up our nitrogen as our plant gets bigger and it's starting to get into that transition phase, where late veg covers some of the transition phase where it's starting to flower. We want to bump up our nitrogen significantly. Our phosphorus is going to go up also. So we're going to go up a full 10 parts per million on our phosphorus, okay? And then potassium, we're going to be looking for also a nice little jump. Come on. And then our calcium also. Come on now. All right, and our magnesium is really just going to go up a little bit. All right, and our sulfur, again, it just needs to be at least that. You know, it's don't worry too much about making sure you have enough sulfur. As long as you have the minimum amount, then you're good. So, okay, make sure I'm keeping up with you guys here. Okay. So now we have our early veg and late veg numbers, and I'll punch in our early flower numbers, which are going to be very similar. Oops. God, I hate this. No, I'm gonna shrink this a little bit. That's better. Okay, so. Let's do this, and let's move into early flower. All right, so we have now our nitrogen is going to stay just about the same. In some circumstances, you will even want to increase your nitrogen. The transition from late veg into early flower, we play it by ear a little bit. We look at the plants, we see how they're responding to everything, and this is the point, and this is the rapid growth stage. That transition between late veg and early flower is when the plant is growing as fast as it will ever grow. Before the flowering stretch and really after that early veg phase is when you have your rapid only in autoflowers, your rapid growth stage. And we want to juice this plant as hard as we can to get the highest yield possible here. This is when it really matters whether or not you give it everything the plant can take. So if your environment is good and if your light intensity is correct, really those two things, pH and all that, but that's kind of part of environment. If that is all good, then you can increase your nitrogen all the way up to about like 275, not 2275, 275. We go up to 300, with our indoor grows, but that's with added CO2. So, if you're adding CO2, if you're really ripping it, you're dialed in, you could go up that high, but really 220, if you stayed there or around 220 to 250, you would be okay. Now your phosphorus is going to increase a little bit as you move into early flower. So at this point, you're going to be at about 75 to 85 for your phosphorus. Your potassium is going to also, you're going to want that at about 220 to 250. And then your calcium, you want it, about also somewhere around right there. And then your magnesium is going to stay the same. We can just for the sake of, we'll bump it up just a little bit here. But really, these small adjustments are you're giving it sufficient magnesium. Really, once you have sufficient magnesium, adding more is not going to do very much for you. Okay, so here's your numbers for early flower, and then we're going to move ourselves into late flower here. Now your nitrogen is going to come back down. Phosphorus. Not even really going to break 100. You really don't need to with phosphorus. Potassium. This potassium number is a little contested. So in all reality, you probably don't need this much potassium. Let me switch back to me for a second. There are studies to indicate that adding this much potassium is just kind of like overkill. There are issues sometimes with potassium uptake in later flower. So adding extra can help to ensure that you're getting the amount that you need. But there have been multiple studies that have shown that this amount of potassium could be significantly reduced, and we are doing our own research currently. Like we're running a few different side-by-sides with more and less potassium to identify whether or not it's, we've gotten excellent results with these numbers. We know that these numbers are like exactly what we need to use in our circumstances and with our commercial clients and all that to hit the highest yields possible. But we're also playing around with the idea of reducing that potassium number just a little bit, just as a heads up. We're always improving. So then your calcium, it doesn't need to be quite as high anymore. Oops. We're gonna go. And then your magnesium can stay right where it is, and your sulfur can stay right where it was. So here are your early veg, late veg. Let me shrink this down. This is down to 13. Your early veg, late veg, early flower, late flower numbers for input PPMs. Now, how do we know that these numbers are correct? How do we know that these numbers are going to get you good results? We do something called, hold on, let me head back to where you guys are and answer any questions. The range decreases as well, 60, 65. Why does it tighten? So dead folk, it tightens because you're doing your best to hit a certain window, and any higher, in some cases unnecessary, some cases can be damaging, and then any lower might not be sufficient. So we're giving you that tighter window to ensure that you're hitting the nail on the head. And if you're not able to get it just there, you get it as close as you can. So like with your nutrient schedule, with your nutrient products, if you're using the Flora series, for example, you might not be able to get it exactly within those numbers with just the Flora series. And what you're going to have to do is pick the element that is the lowest possible number. So here, that's what the next thing that we're going to do is, doesn't the higher potassium help the usage of calcium? They do interact, dead folk. It's not quite as black and white, but it's over 2,000. Yeah. So let me make sure that I'm current with you guys because last time it was lagging. Perfect. Yes. So now, for example, let's just choose our early veg, okay? So we know that we need 180 to 220 parts per million for, when your plants are in just past the seedling stage. So we know that we have, let's see, 2% nitrogen in the flora grow. We have 5% nitrogen in the micro. Let me pull this up for you guys so you can see it. Perfect. So now what we need to do is, we've already done the math and we know that if we add 2.5 milliliters per gallon, that we get these numbers. So we are going to just increase that application rate until we get to the number that we want to see for nitrogen, and we'll check to make sure the other numbers are not too high as well. So let's see, if 2.5 milliliters per gallon gives us a total of 46, then, because remember, we added all the nitrogen together, we added all the phosphorus, we added all the potassium. That's what this is here. So if that at 2.5 milliliters gave us a total of 46, then we're just going to do 200. Let's just start at 200 divided by 46 equals 4.3 times our 2.5. So how many times does that 46 fit into our ideal number of 200? It's 4.35 times 4.35. And then you're going to multiply that by your current application rate. So that is 2.5. And that gives you 10.875, or about 11 milliliters of the nitrogen. So now we're going to check our other numbers and see if we're way off. And I'll tell you what we do if we are. So moving into, so if our desired nitrogen, here, let's do this. We want our nitrogen at about, let's just use one number here. So 200. We want our phosphorus at, let's make it easy, 50. And we want our potassium at 180. Okay, let's just make these numbers nice and simple. And okay, so now we can look up here and see that if we multiply this by 4.35 to get to this 200 number, then we're also going to have to multiply, if we're adding them all in the same amount like it recommends, then we're also going to have to multiply the phosphorus number by 4.35, and that would be 39 times 4.35, 170. We're way too high on phosphorus. So there's no way that we could just keep it as equal amounts and multiply it up to get to the nitrogen number. So the next thing that we have to do is, now we know that phosphorus, which is usually the case, is our limiting factor. So if we want it to be 50 and we're already at 39, which is a combination of your phosphorus in your bloom and in your grow, which is 6.6 parts per million. So now we know our grow has 13.2 parts per million of nitrogen, 6.6 parts per million of phosphorus, and it has 39.6 parts per million of potassium. So we play around with the numbers until we get to our desired amounts. And I'm sure there's make sure I see comments coming in. Doesn't higher potassium, hello, I saw that already. Going to have to watch this again without ripping my fresh flower rosin. Yeah. It's a little bit dense. It's a little bit dense, and this is more of like a resource for people who want to, come back, and if they're ever interested in producing a feeding schedule with the nutrient line that they're using, then this is here. Watch it a few times. Try to follow along as you're doing it. But what the idea of what we're doing next here is we know where we want to be with our PPMs. We know the total PPMs, per milliliter of each of the 3 elements that we're adding. Now we play around with the numbers, add a little more of this, add a little less of this, play around with it until you get to those ideal numbers. And if you want me to do it here on the stream, I can. We're kind of running out of time. That's something where when I get off the stream here, I can do all the calculations, add it to the document, and kind of put a little explanation of, okay, we ended up adding more flora grow, we kept the flora bloom the same, and we added a little bit more micro, and that got us to where we needed to be. But oftentimes you'll end up right where you need to be, but your potassium's a little bit too low. And when that happens is sometimes you need to go and get an extra potassium product, because if you add more of whatever your flora grow or your flora product to get more potassium, you might go way higher on your phosphorus than you want to. You don't have anything that's just potassium, so you can't increase just that without increasing other stuff. And sometimes it's often the case where you'll have to add another element because you're too far away from the ideal numbers. Oftentimes, though, you'll be able to get just about right to those numbers, a nice balance where it's like, okay, close enough to 180 here. I'm close enough to 200 here. We're within that range where this is the best we're going to do with these 3 products. So that's kind of the explanation of how you take your calculation for PPMs, your ideal PPM inputs, and then use those to create inputs for your nutrients in milliliters per gallon to get to those PPMs. Can I speak on Brix levels if possible? I know it's not nutrition, but it isn't an indication of health. Isn't it an indication of health? Aphids can't process the sugar, so they don't pressure plants. Yeah, so Dead Folk is talking about Brix levels, which is the level of sugar in your plants. Brix levels are used in all sorts of different agricultural crops, and they have little Brix readers where you put a little bit of sap on like a little... My microphone thing just fell off, of course, so I'm going to have to hold it for the rest of the thing. My desk does not have like a big enough lip for the microphone to clamp onto it, so it's like always precariously perched on the edge. So I will hold the microphone. So Brix levels, those you can use a little device where you put a little bit of sap, and I don't remember if you dry it out or if it measures it wet, but it can measure the Brix levels just based on like the optics of the sap, and it is so Brix is something you measure throughout the life. Yeah, I mean measuring Brix can be very helpful for identifying if you're able, like you said, it doesn't even register to a lot of pests if your plants are high enough Brix levels. They don't smell it. It's not the same. They don't go after plants that have high Brix levels. And now a lot of this is semi-theoretical. This is not applied in most cannabis cultivation circumstances, scenarios, because cannabis is not a plant that produces a ton of sugars. So Brix levels are spoken about a lot more in food crops, grapes and corn and that kind of stuff. But with cannabis, it's lower on the rung of things that are considered to be important with commercial cultivation. It's not something I have like extremely deep knowledge on how to increase your Brix levels infallibly, but it is something that I have done some reading on, and I am aware that it definitely makes plants less attractive to insects. I know that how to read Brix levels, but as far as like how to increase your Brix levels to achieve that, I'd have to do some research and get back to you. So let's see. How do you calculate PPMs if the nutrient company use grams instead of milliliters? That is a different calculation. I'll add that to the document. I can do that. But in reality, it's very similar because 1 gram of water equals 1 milliliter of water equals 1 gram, right? Am I remembering that correctly? Let me tell you right now. Right. Yes. Okay. I don't know why I second guessed myself there. So 1 milliliter of liquid water, equals 1 gram, and your specific gravities can be higher or lower, but generally you can. And also, you know what? Here's what I'm gonna do. Let me see if I can just put this on my lap for a second. Prop it up right here. There is a great calculator for that. There's an online calculator for that one. Give me 1 sec. I will pull it up, and I will put it in the chat. Actually, you know what? I can do it on the other one. Yeah, that's a better idea. I'll pull it up on the one that you guys can see. Okay. Let me do this. So you guys are still seeing that, right? Yeah, okay, cool. I'll pull it up over here. Okay, here we go. All right. So you guys can see this now. Let me adjust the size of the window here so that you can see what I'm doing. I think I'm gonna need to. Is it Alt? There we go. Okay. So now here, this is the website. You gonna be able to see that? Can I scroll all the way up? Okay, here. I'll fix this. Give me 1 sec. So here is the website called omnicalculator.com. If you just search for this, Omni, OMNI, and then you do water soluble fertilizer calculator, you can get this calculator. And I'll show you how to use it real quick. I've used this a million times, and it is very helpful for if you are indeed using salt nutrients. Not organic, not organic top dress or anything, but pardon the noise. Hold on, let me move the mic away so I can bump the mic thing back onto the desk. Okay, maybe that worked. It's connected. Okay, we are good. So this calculator, here's how it works. Let's just, here. Let me go back to stream. Who was asking me that? Mattman, which nutrient line do you use? The one when you're asking about grams instead of milliliters. So obviously it's a soluble salt nutrient. Which one is it, like crop salt or what? If you have a preference, otherwise I'm just going to choose one, like Jack's. So here, I'll just put in Jack's 3, 2, 1. So we've got here, let's just do 15, 12, 26. That's their part A. So I don't know exactly what their recommended feeding rate is off the top of my head, but I'll just wing it here. I'll just put like 5 grams. So kosher, I don't know that one. I could look at. I could do it after this real quick if you really want. But all right. So we had what was it? 15, 12, 26. So we've got nitrogen is at 15. Phosphate is at 12. And you're at potassium is at 26. I like to do the elemental percentages here. Just because that's where you can add your calcium, your magnesium, your iron, everything. So this calculator is great. So now I just added in my NPK, just simple, nice and simple, just the NPK. And now here, so we've got switch this to Um, fertilizer rate. Here we go. Switch to gallons. US gallons. And we want to do 1 gallon. So we're doing grams per gallon. Let's just say Jax wants us to add 5 grams per gallon, okay, of that part A. That's part A, right? Yeah, of their part A. That's 15, 12, 26. All right, so I added in 5 grams. I switched just to 1 US gallon. And now we've got, it doesn't give you more, right? Yeah, that's it. Okay. So now we've got our milligrams per liter, which is the same as your PPMs. Milligrams per liter is just another way, essentially, of writing PPMs. It can be slightly different in certain circumstances. Say that 10 times fast. But it for right now is fine. So I just, off the top of my head, I just winged it and chose 5 grams. And look, our NPK is actually really solid. You know, that just gave us 200, 285, which is a little high on the potassium. This is like good for like moving into late flower. This is a really nice NPK for moving into late flower. So does this answer your question, Matt? Play around with this calculator. You know, like I could go in and pull up their full, see if I can get a picture of their guaranteed analysis here, and it's actually readable. Not a chance. So yeah, no. Switch back to me real quick. So yeah, you can go to your guaranteed analysis. You switch to the elemental percentage on the Omni calculator, and you just type in everything, and you type in your number of grams. Switch it to 1 US gallon. Make sure it's switched to 1 US gallon, and then you just go ahead and punch it in, and it gives you all your PPMs. It's just as simple as that. There used to be a good calculator for that to do it for the liquid. We didn't have to do the calculation. Like I said, we are working on getting a calculator up on the speedrun site, so you don't have to do all this math. You just punch it in. I'm actually also working on an AI like chatbot with ChatGPT where you can give it the stuff and it'll do it for you. Right now, it's only available if you have the paid version of ChatGPT. So if any of you guys do have the paid version of ChatGPT, I can send you a link for the chatbot that I've done a little bit of training with on. You know, it's called the Speedrun Grow Advisor, and it'll calculate PPMs for you. So, okay. Where are we at? Last thing I want to talk about is plant tissue analysis. Plant tissue analysis is the reason we know all of these numbers, that we're confident in all of these numbers. So plant tissue analysis is taking I'm even going to pull up a little plant tissue analysis study for you to end the stream here. So this is done across all crops. This is something that is just like considered to be the standard for identifying whether your plants are well fed, removing the guesswork. You're not looking at yellow leaves or this plant's a little droopy. You're not looking at anything like that. You are just looking at your actual amount of your nutrients in your tissue. So this is North Carolina State University, and they have a big hemp program there. Okay, so this is their hemp leaf tissue nutrient ranges, refinement of reference standards for floral hemp. Floral hemp meaning like CBD hemp, not fiber hemp. Which is the same as cannabis. You know, like the nutrients are all very similar. It's hemp and cannabis are the same species. So definitely, I'll post a link for this if you want it. Here, I'll pop it into the chat. And you guys can read through it if you want. But generally, what they have here is, here's some sufficiency ranges. So they're showing you an example of your nutrient concentration in your tissue and your number of tissue samples, I guess, here. And then they're identifying the peak of the graph is where you have a sufficient amount of that nutrient in that many tissue samples. So this is here we go. So here's 3 different studies, and we could pull up these studies individually at some other time, but can you, here, let me fix that so you can see it. How far over does it go? Okay, so here's 3 different studies, one 2014, one 2020, and one 2019, and this is what they found to be their ideal percentages of nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. And these can be converted into parts per million, but this is how it's expressed to you when you get, so this is when you have a leaf tissue sample, you send it to the lab and you get it back, you're going to get a number like, 3.2% nitrogen. And you kind of just have to know, based on the sufficiency ranges that you've laid out, what is ideal. And now the question, do I agree with all of these numbers that they've come up with? I would say the one here that is the most accurate, probably this middle one, Kalinowski et al. I like their take on this. Calcium, really 1.5 is way too low. Like that, you would never want it to be that low. Potassium, yeah, these numbers are pretty good. Your phosphorus numbers here are good. Yeah, nitrogen 3.5 to 5. So you can get your plants analyzed. You send them to the lab. You send 20 to 25 leaves in per sample. They run that sample through their, I think it's gas chromatography or liquid chromatography or gas mass spectrometry or liquid chromatography. And they give you back your mineral content in percentages or parts per million with the micronutrients in your plant tissues. Certain nutrients build up, like nickel and iron, those heavy metals build up in your plant tissues. So as you move later into the grow, it's less necessary to load them with as much because you've already built up a lot in the tissues. You can have your silica tested as well, or silicon tested as well. And this is how we can identify, okay, we're adding 220 parts per million of nitrogen. We have taken 30,000 leaf tissue samples in the course of our history of doing agronomy with cannabis clients and our own plants. And we've also done a ton of research on other people who have done sufficiency ranges for cannabis for leaf tissue. So we have all of our leaf tissues tested. We say, okay, we know what we're adding. We know what's coming up in our leaf tissues. What do we have to adjust in our inputs to get to those ideal sufficient ranges that we want to see, just like these people put together? And we have our own sufficiency ranges for all the micro and macronutrients, and we have our own input amounts. Now, they're different for each environment. If you have CO2, if your light intensity is higher, if your environment is good, your humidity is high, your humidity and your temperature are good for the plant. Your VPD is good, and that means your transpiration is good, and the plant is running through the nutrients at the pace that you want it to. All of those things play in to the ideal inputs. But when everything is good, when all of those numbers, when your pH, when everything lines up well and you're dialed in, those input numbers that I wrote on the document there, those are what you want to see. That's what you want to add in order to get to your ideal numbers. Joe, in all stages of growth, no, not necessarily. The reason that you see a range 1.5 to 5.4, I think it was, is because it increases throughout. Like that's for the full, it's a range, so you want it to be a little lower at the beginning, maybe a little higher at the end. So it gives you the full range, and then your ideal numbers at each growth stage are going to be within that range. Does age of leaf play into the... Yeah, absolutely, dead folk. So when you take leaf tissue analysis samples, you take a sample of a most recently expanded fan leaf. So your fan leaves, you've got your meristem, your leaves, so you can see my hands here, your meristem, your leaves begin to they start pointing upwards. They begin to kind of grow outwards, and they flatten. As soon as they become flat, they're fully expanded. They are, they look like a mature fan leaf, but they're not the big fat ones that have been around on the plant since the beginning. They're recently matured, recently expanded fan leaves. And you collect 20 to 25 of those per sample, which can be a mixture of strains. It can be a bunch of different plants that are all the same strain. It can be a number of different ways. You can do it all off of one plant if you want to get a tissue sample from one plant if it's big enough. Or you test your whole field. So you just take from random places across the whole field. Doesn't matter what strain. But you can track it every week and make sure that you're hitting those sufficiency ranges for that particular week all the way till the end. And that's how we maintain getting the highest yields possible, is by tracking our nutrient content in our plants all the way through. So that's plant tissue analysis. That's the science of it. That's how we've created a successful system for cultivating autoflowers on a large scale. And we are doing everything that we can to miniaturize that system for home growers. And we want to make sure that's what the idea of full auto was, is to take everything that we learned from doing this on a commercial scale and shrink it down into a product where you don't have to do any math. You just add it, shake it, do it. And we've done all of that for you. All that stuff that you just saw went into full auto. So it's a lot of love in that. Okay, guys, we're over an hour. Any questions at the end here? If any of you guys pays for GPT-4, let me know, and I can give you a link for the Speedrun Grow Advisor that I've made. It's like my own little GPT. I can give you a link for it, and you can help me train it a little bit. Other than that, yeah, I think we covered it. That was a lot. Two long streams. I'm going to try to do my best to pull some useful clips out of these 2 streams and make a shorter video. And I might even record a little bit more and add to it, just so that it all kind of comes together and makes sense. But yeah, guys, I think that's where we're going to wrap it up. We've got a drop coming pretty soon. I think I said at the beginning of the stream, we got a drop coming that's probably going to be exclusive on Multiverse. Anybody who has pictures of the Ice Latte crossed with Jehovah's Witness, hit us up with them, because we were going to pick some people's pictures to use for that strain. That's one of the ones that's coming out next. We're going to bring back Rainbow Six. We've got, we got a lot. We got testers that are going to be, available soon, which are, like strains that have not been run by crosses that we've made that we haven't run yet. So yeah. All right, guys. Happy Monday. I will, actually, there's some videos coming out this week. The tent's running. We got, I posted a picture on Instagram of the roots that we did, that we had when we transplanted. Seedlings are about yay big. You're going to see a video of those this week. And how we're setting up the tent. Can I get a witness? Yeah. So, okay. Little Mac, if you want some suggestions, I would say the Granite Haze is a classic. Can't go wrong with it. And then I really like, my personal preferences are like the Sapphire crosses. So Rock Slide, Holy Fire, Sapphire itself. Yeah. So, and Frosted Cheerios is excellent. Okay, guys. All right. I will, I'll talk to y'all soon. Have a good night. Where's my end stream button? There it is.